So welcome back everyone to UCSF's Cardiology Grand Rounds. My name is Ram Venkateswaran, and I'm one of the third-year cardiology fellows stepping in for Layla, who's busy today. I'm honored today to present Dr. Lior Gepstein. He is the director of the cardiology department at Rambam Healthcare Campus in Haifa, Israel, where he is joining us from today. He completed his medical and PhD degrees at the Technion Israel Institute of Technology prior to coming here to UCSF for a 2-year fellowship in electrophysiology. And currently he is a professor of physiology and medicine in cardiology at Technion's Faculty of Medicine and holds the Edna and Jonathan Sonis Chair in Tissue Engineering and Regenerative Medicine. Dr. Gepstein's research interests are vast. During his training, he was involved in the development of the CARDO system, which is the state-of-the-art electroanatomical mapping technique used in many EP procedures now. His research group notably was among the early pioneers in developing techniques for the differentiation of human stem cells into cardiomyocytes. Presently, his research focuses on stem cell biology and the development of novel gene and cell-based therapies for treatment of various cardiovascular diseases. Dr. Gepstein has received a number of prestigious awards for his achievements in research, including the American College of Cardiology's Douglas Zipes Distinguished Young Scholar Award, the European Society of Cardiology's Outstanding Research Achievement Award, and the Murawski Award from the Israel Cardiology Society. He also has been elected to the Israeli Young Academy of Science. Recently, he gave a talk to our EP group, and we're excited to welcome him back to the division's grand rounds to learn more. With that, Dr. Gepstein, welcome. I'll hand it over to you.
Thank you, Ram. It's great to be back in UCSF, if not in person, remotely. As you told everybody, I spent almost 2 years, many years ago. It was probably one of the most fun we had in my career. So what I'll try to do today is talk to you a little bit about the work we've been doing over the years in trying to use advanced biological methods, such as you see in such as you can see in this slide.
Control alone, and he's okay doing that.
Can you see it? Can you see it?
Yep, we're all good. We can see your
Okay. So Nature has a competition every year about the biological method of the year. And the advances we have in biology is really stunning. And the question is how we bridge the gap from these amazing technologies in the lab to use them to actually help us understand disease and maybe develop better technologies. And we've been trying to use some of these technologies in the lab over the years, mainly in the field of cardiac electrophysiology. So the technologies that I will mainly discuss with you will be human pluripotent stem cells, tissue engineering, and optogenetics. And the first technology that we used almost 2 decades ago, a little bit more than 2 decades ago, were human embryonic stem cells. These cells originated from the early in vitro fertilized embryo. They were derived, as I told you, more than two decades ago, in collaboration of my hospital, Rambam, together with University of Wisconsin, and they have the capacity to propagate indefinitely in culture while having the potential to become any cell type. And a little bit more than 20 years ago, we were the first to use these cells to differentiate them into cardiomyocytes, as you can see here. And this is like a historic movie. This is the first human heart cells that were generated in the lab. And we can hold these cells forever. I've been showing this slide for more than 2 decades, and they still beat in my slide. But the longest we've been holding them is for more than a year. So over the years, we tried to see if we can use these cells for infarct repair and treatment of heart failure. And the first question was, can these cells integrate with existing heart tissue, electrically and mechanically? And we ran them through several studies, both in vitro and vivo. This is one of our earliest in vitro study, and we put our human cells next to rat heart cells. And as you can see, they started to beat in synchrony. And when we're recording electrical activity, we show that actually this is because they integrate and generate a single electrical functions in situ. After showing that they can integrate both in vivo and ex vivo, and one of the studies was actually done in Jeff's lab when I was at UCSF, we then went to see if they can actually improve the mechanical performance of diseased heart. And again, we used a number of models over the year. This is one of our earliest studies in which we did an infarct model in rats, transplanted the cells into the infarct area, and looked at their functional shortening over time, and noted that in contrast to control group, where we only injected culture medium, that we can see significant deterioration over time because of the remodeling after an infarct. Animals that were injected with our human heart cells actually improved their function. So this was many years ago. So what was the problem in using embryonic stem cells? One of the problems is that you will not be transplanted cells that are autologous to the patient, and therefore you would expect immune rejection. And this also does not allow you to generate patient-specific models. The solution to this came in 2006 when Shinya Yamanaka, a Japanese researcher who also works at Gladstone Institute, reported on the development of the induced pluripotent stem cell system, or iPS cells, in which he can take adult cells from skin or blood and reprogram them by a set of transcription factor to become embryonic stem cell-like cells, which he called iPS cells. And these cells can then be coaxed to differentiate to any cell type, and then can be used for autologous transplantation strategies and for generating patient-specific in vitro disease models. And this turned out to be a really robust technology. We were one of the first, again, to take his technique, generate iPS cells from patients, and then use our differentiation scheme to generate the patient-specific heart cells. Okay, so now we can make patient-specific heart cells. The question, can you make enough of them? And in adult heart, there's about 4 billion cells. And in an infarct that results in heart failure, we lose 1/4 of them. So you need to actually regenerate close to 1 billion heart cells. And our initial differentiation system were not very efficient. Over the years, we became better by trying to mimic what happens during early development. And by using a number of differentiation techniques, we can now be very efficient in our differentiation, where we get more than 90% of the cells becoming heart cells. And we can get something like 1 billion of cells with a cost of about $1,000 or even less now. So we can make enough heart cells. So what is the next frontier? So first generation clinical studies will probably involve transplanting cells directly into the infarct. But next generation of studies may include the tissue engineering concept. The idea is that our organs are not made only of cells. They are made of cells that are growing on extracellular matrix that behaves as a scaffold. And tissue engineering can allow us to mix cells together with polymers to generate tissues or organ-like structures. And over the years, we had several protocols to do this. Our first protocols were actually a collaboration with the Zimmermann group in Germany, Zimmermann and Heschenhagen, in which we take collagen together with heart cells. You mix them in a chamber that you make any shape you want. In this case, we want to make ring-shaped tissues. We then put it on a stretcher. It's like lifting weights for the cells. They hypertrophy and generate a tissue that starts to contract. You can see these spontaneously beating rubber band-like structures that can generate a significant amount of force. And we can do it also from iPS cells. We call these beating calamaries, that we use them not only for transplantation purposes, but also as an in vitro model to study mechanical force formation by human heart tissue for drug screening. One of the problem in tissue engineering is the thickness of the muscle that you can make. because it's dependent on diffusion of oxygen. And if you make it too thick and transplant it, the center would probably die of necrosis. So one of the projects we've been working over the years is to try to generate an already pre-existing vasculature within the tissue. So here we combine heart cells together with vascular precursor cells, and we can generate blood vessels within the engineered tissue together with the heart cells. And if you then transplant this engineered tissue on a heart, in this case a rat heart, you can see that many vessels form in the engrafted tissue. About half of it comes with the cells, with the tissue, because they are stained positively for human, and about half are ingrowing rat vessels that grow into the scaffold, and the human vessels become functional, and as you can see, perfuse red blood cells, thereby improving the vascularization of the graft. Okay, so we showed you that we can make engineered tissues in predetermined shapes, but this is not really the shape of the heart, both microscopically or macroscopically. So to try to target this, we're using what we call a decellularization-recellularization process, in which we can take a heart from mice, rats, pigs, etc., and then decellularize them by enzymatic reaction, get rid of all the cells, so you remain only with the extracellular matrix in the shape of the heart. And then you can reseed this heart with human cells. One example, in this example, we take part of the ventricular wall, we recellularize it, as you can see, with human cells, and then we can use it for transplantation as a transplantation patch in these experiments. So this is one approach. Using patches in general will require you to transplant this using surgery. And interestingly, this approach by my collaborator, Wolfgang Zimmermann, has already reached clinical studies in Göttingen. They've already transplanted this patch, which is about 10 centimeters square, that includes about 1 billion heart cells in about 5 patients, I think, during surgery. An alternative approach may be, because we of course, as cardiologists, don't want to give work to the surgeons, is to try to do something minimally invasive to inject directly to the heart. And here we're using an in situ engineering approach in which we inject both a gel that can polymerize inside the heart with the cells, and therefore the tissue actually is formed within the heart. So there's many ways of how to form this polymerization. Here we're injecting the gel together with the cells, polymerizing it. And as you can see, when we inject only saline, there is a typical remodeled infarct heart, which is the scar is very thin, lacks cellularity, and there is significant dilatation, remodeling of the chamber. When we inject the cardiomyocytes with the polymer, you can see that the anterior wall remains thickened, there's much more cellularity, and there is less remodeling. So if we compare injection of saline, which we see adverse remodeling, to only polymer, which actually prevents some of the remodeling, cells alone, we can see that the best is polymer with cells. Okay, so we talked about injecting the heart with cells alone or as a tissue. The next question is, which cells are we making? So when we look at the cells that we're making from our iPS cells, we see that we're making a mixture of different ventricular, atrial, and pacemaker-like cells. And you know that the heart has several different types of cells. Beyond these chamber-specific myocytes, we also have conduction system cells. We have non-muscle cells, such as epicardial cells, endocardial cells, cardiac fibroblasts, and so on. So the next actually challenge is to try to develop differentiation protocols that will give rise to a specific purified cell population of the heart. And over the years in the lab, together with collaborators like Gordon Keller in Toronto, we've been able to generate protocols to get almost any cell type of the heart. This is done by actually imitating what happens during normal development. It turns out that most decisions regarding when the mesodermal cells become heart cells very early on, they both decide that they become heart cells, and already very early on, they decide which heart cells they want to become. So by manipulating the growth factors during differentiation, you can actually make specific heart cell types. And for example, in this study, we were able to generate protocols that generate only ventricular cells or only SA node pacemaker-like cells. The ventricular cells, of course, we want for transplantation studies in the ventricle for heart failure treatment. The SA node cells we can potentially use as a biological pacemaker. And in this study, we show that if we take these SA node cells, put it on ventricular tissue, these SA node cells can actually pace the ventricular tissue. And if we inject them into the apex of the rat's ventricle, we can optically map them and show that during normal sinus rhythm conduction at the ventricle that starts at the red area and then propagates to yellow, green, and so on. You can see that during normal sinus rhythm, conduction initiates at the septum, not at the site where we transplanted our cells. And this is not surprising because the heart rate of the rat is faster than our human SA node cells. So when the heart rate is normal and fast, our pacemaker cells do not kick in. But when we create AV block by pharmacology induced AV block, you can see that our cells start to kick in, and now the earliest activation site shifts to the area of cell transplantation. So potentially we can use this approach as a biological pacemaker approach, and now we are doing this studies in pigs. Okay, so we talked about using these cells for regenerative medicine. I told you that these approaches are now already entering clinical trials. But what more can you do with the IPS approach? One attractive approach is to try to use them as in vitro models to study genetic disorders. The idea, if one has a cardiomyopathy or in channelopathy, if you take his cells, reprogram them to IPS cells, and then generate his heart cells in the dish, the hypothesis is that these heart cells will portray the patient's specific abnormalities because they're supposed to have the same genetic material as the patient. So to test this hypothesis, a few years ago, we had a young patient who came to our ICCU after she had cardiac arrest in the street. When she came in, she had these storms of torsade de poids. We saw that she had a long QT interval, and we learned that a number of family members died at a young age. So we screened the family and identified a point mutation in the KCNH2 gene, encoding a potassium channel, which is responsible for the long QT type 2 syndrome. You're all familiar with the long QT syndrome. So we wanted to see whether we can use this concept of IPS modeling to generate a model of that specific patient. So to this end, we took some cells from the patient, reprogrammed them to generate IPS cells, then made the patient own cardiomyocyte in the dish, and started to study it. At that time, we still did not know how to make specific heart cell types. So our differentiation resulted in a mixture of cells that had ventricular, atrial, and nodal phenotypes, if you look at their action potential. So when we studied using patch clamp IPS derived from a healthy volunteer, you can see that we can get the majority of the cells having a ventricular phenotype with a ventricular-like action potential. When we did the same with our long QT patient, you don't have to be a cellular electrophysiologist to see that the action potential duration is prolonged in the ventricular and atrial cells of our patient due to mutation in this potassium channel. Not only that, but some of the cells developed early after depolarization and triggered beats, and even a series of trigger beats. So you had a young patient coming with a VT storm, a long QT syndrome, and then you could make our own heart cells in the lab portraying the same phenotype. So now you can study the mechanism by which the mutation causes the problem, and potentially test specific treatments on the patient's own cell. Here, for example, we gave a drug that opens an alternative potassium channel, the ATP channel, by giving pinacidil. And you can see that by opening this channel, we were able to shorten the action potential duration and eliminate the early after depolarization in the cell. So to test whether we can actually guide a treatment in our patients, we did another study. In this study, we created an IPS model of a different genetic disease. This disease is called CPVT. There are two types of CPVT. CPVT is, again, an inherited arrhythmogenic disorder that can cause sudden death in young individuals when they exercise. The most common inheritance is autosomal dominant due to a mutation in the ryanodine receptor. But we are taking care of a large Bedouin family that has CPVT due to an autosomal recessive disorders called CPVT2. And the mutation was actually described first in this family in the world. And this is due to a calciquestrin mutation, which the calciquestrin is the main calcium buffering protein in the SR. The idea is that probably to the lack of this calciquestrin, there is calcium drifting out of the SR during diastole, which introduces a cascade of events that results in this arrhythmogenic bidirectional VT. So every time these kids in the family exercise, they develop this arrhythmia, and so this is a very malignant disease in this specific family. When we implant ICDs to them, it's actually not also optimal because if they get an arrhythmia, they get a shock, the shock is painful, then it induces a burst of adrenaline, they get another shock. So sometimes they come in with, with 30 consecutive shock because of VT storm. I remember we have one of the boys is in adolescence, he has all these hormones. Every time he sees a nice girl, he comes up with multiple shocks to the hospital. So it's not a great quality of life, although they are treated with beta blockers. So we wanted to see if we can treat them better. So we made some iPS cells of these patients, of this family, transformed them into heart cells and studied their calcium handling properties. And we noted that these cells are very arrhythmogenic with a lot of calcium release events during diastole and oscillation. So they're extremely arrhythmogenic. So we tested which drugs can be beneficial for these patients. And we noted a couple of drugs that can be beneficial by reducing arrhythmic activity in the cells. For example, flecainide, which was suggested as a treatment for CPVT type 1, was very good in eliminating arrhythmia. And also carvedilol was very good. So we tested this on the patients. And first, this is the patient's cells. You can see that it stopped their calcium handling abnormalities. When we took the same patient from which we made the iPS cells, and put him on a treadmill, you can see when he just starts to exercise, he has this ventricular ectopy. But when we give them flecainide, it prevents the generation of ectopy. So flecainide was found to be a very good treatment for this family, and many members of the family are now treated by flecainide. This was based both on our results in the IPS model, but also because other people have demonstrated that it's beneficial in CPVT type 1. Okay. So the two disease models that I showed you, you can actually model the arrhythmia at the single cell level. But what if you want to model something more complex? For example, atrial fibrillation. Why would you want to model atrial fibrillation? We all know that. First of all, it is a huge clinical problem. Second, the mechanism underlying atrial fibrillation are probably multiple, and we want to learn a little bit more about this. And third, there is increasing evidence that genetics play a role in atrial fibrillation. So we said, okay, let's see if we can try to study this in a dish. So this we have to do this, we have to face two problems. First of all, we have to make atrial cells to study atrial fibrillation. And second is that arrhythmia, like reentrant arrhythmias, cannot study at a single cell level. You need a tissue to study reentry. So we try to tackle both. First, we developed a method to differentiate the cells to atrial cells, and we do this by protocols to derive only ventricular cells or atrial cells, and you can see the atrial cells with the appropriate atrial action potential properties and the presence of atrial specific channels in the cells, like the acetylcholine-dependent potassium channel. So we have atrial cells. Now the next question, can we make a tissue? So we started by looking at relatively primitive models. So we started with a 2D tissue model that you can see here. This is composed of about 1 million of atrial cells. And even by eye, you can see the propagation of the mechanical force propagation in the tissue. This is how it looks under the microscope. I don't know if it looks well via Zoom, but you can see the contraction of the tissue. We then wanted to study them, so we generated an optical mapping system designed to study cultures, and we can study the fluorescence of the tissue after loading the cells, the tissue with voltage-sensitive dyes that change their fluorescence based on changes in the membrane potential. And as you can see, we could study the propagation of electrical activity in our tissue. You can study conduction, repolarization, and so on. We can also do programmed electrical stimulation like we do in the EP lab. And we can induce, I don't know, this again, I don't know if you can see, we can use rotors or spiral waves in the tissue. I'm not sure it's projecting well over the Zoom. We can now study these rotors. We could look, for example, on environmental or genetic factors. What do they do to the tissue properties? So we can look, how does conduction look at baseline? How is arrhythmia induced? Here we are pacing fast, and we can see that a rotor is being developed. I hope you can see this because it's a little bit slow at my end. We can look how the rotor is maintained and how it can break to daughter wavelet during fibrillation. Okay. So we now have a tissue. What else can we do? The next thing we introduced is involving another biological technology that won the Nature Method of the Year and actually won the Nobel Prize. This is CRISPR-based gene editing. The idea is to use a molecular scissor approach to cut the DNA at a specific site. You can then correct a mutation, introduce a mutation, and so on. So to test the feasibility of using this approach together with the IPS modeling, we studied another disease using our models called the short QT syndrome. The short QT syndrome is also inherited arrhythmogenic syndrome. It turns out that if you have a long action potential duration manifested in the ECG as long QT is bad for you, but also if you have a short QT, a short action potential duration, can also be arrhythmogenic. And the mutation leading to the short QT syndrome is usually at the same genes as the long QT syndrome, but causes an opposite effect. So if in the long QT syndrome, mutation in potassium channel, like the one I showed you, causes a loss of function endpoint, in the short QT syndrome, in the same potassium channel, we can see mutation that result in a gain of function mutation, resulting in too much of a potassium current, which shortens the action potential. So we studied a family in collaboration with a group in Germany that has the short QT syndrome and history of atrial and ventricular arrhythmias. We made IPS cells from the family and studied their action potential duration using patch clamp. You can see that here, the healthy control looks long, while the short QT syndrome looks significantly shorter. So we could see that action potential is much shorter in the short QT syndrome, and we also lose the ability of the cells to change their action potential duration versus rate, in contrast to the control cells. So at the action potential level, we have shortening of the action potential duration, but to study arrhythmias in the short-term QT syndrome, again, you need to have a tissue because there's nothing like EADs that we see in the long QT syndrome. So again, we use the short QT syndrome we generate a monolayer of short QT derived heart cells, and we can easily induce arrhythmias in our short QT syndrome versus healthy control. You can see that more than 80% of the short QT ventricular tissues generated transient arrhythmias, while only 30% of healthy control. We then wanted to see what is the cause of generating arrhythmias and why they are more sustained in short QT syndrome. The cause that it's easier to induce rotors is because of the shortening of the action potential duration, that it's easier to generate arrhythmias because of shortening of refractory period. Why does the short QT syndrome is more sustained, the arrhythmias were generated? So we took advantage of the fact that we can generate rotors both in some of the healthy in the short QT syndrome, and we studied their electrical biophysical properties of these rotors, and we saw that they have difference in frequency of the rotation, differences in the curvature of the rotors, and differences in the meandering of the rotors. You can see that the short QT rotors are much more stable in space, while the healthy are less stable and move around, meaning that there's a greater chance that they will collide with obstacles or another rotors and terminate. So we have ideas now why short QT syndrome it's easier to generate arrhythmias and why they are more sustained. We then wanted to see whether we can correct the mutation. So we use CRISPR-based gene editing to correct the mutated allele. And we generate what is called an isogenic control line, which is identical to the short QT syndrome iPS cells in terms of the genetic background. The only difference is that the mutation that exists in the short QT cells but does not exist is converted into wild type in the corrected short QT cells. So this is actually the optimal control line. And we saw that action potential duration in the short QT syndrome, which is short, is corrected in the corrected short QT syndrome. This actually proves that indeed this DNA change was the cause of the phenotype that we saw. And if you look at the arrhythmia, we cannot generate any arrhythmias in the corrected line anymore. We also use this model to try to find which therapy will be best for these patients. To this end, we tested the effects of different drugs. Since the mutation results in gain of function mutation, too much potassium current, we thought maybe by giving something that blocks directly the potassium channel, we'll be able to correct the phenotype. So we tried sotalol, which blocks the hERG channel where the mutation is. And interestingly, it did not change the action potential duration of these cells. So despite using a potassium channel blocker, action potential duration did not change. And this we believe it because sotalol binds to the potassium channel at the area where the mutation exists, and we think that the mutation hinders the binding of the drug to the molecule and therefore prevents its activity. So we tried a different drug, quinidine, which is more dirty and affects different channels. And quinidine actually prolonged action potential duration. And this was enough to prevent the initiation of arrhythmias in these models. So quinidine is a good drug for this type of inherited arrhythmia. Okay, so I showed you that we use simple 2D models to start to study reentry. But when we want to go to real life, the 3D anatomy is much more complex than what we are trying to simulate in vitro. So this is how an atria look, and you want to consider all these anatomical issues when you want to model something like atrial fibrillation. And similarly, if you want to model something like scar-related VT, you have to think on all the pathological substrates that exist. For example, the presence of surviving tissue in the border zone that allows the generation of VT. in a healed scar. So the next stage, we're trying to move from a 2D models to 3D, more clinically relevant models. And I told you one way to do this is to use decellularization, recellularization approaches. Here, for example, we are taking the atria, decellularizing it. So you see now we're staying from a son crichome. There's no cells left, but the anatomy is preserved. And then we can recellularize it with the human atrial cells. So we are trying to make atrial tissue with the macroscopic and microscopic structures of the atria with human atrial cells. We are now trying to make a 2D model by opening the atria and making it a flat surface to try to optically map the atria. Here you can see that the cells beat and they actually align along the fibers of the extracellular matrix that gives them somewhat of the microscopic structure of the tissues that we're trying to model. We can now load these cells with voltage-sensitive dyes, and we can actually induce, using pharmacological manipulation, like we do in some animal models, induce atrial fibrillation like activity in these models. So this is one approach to generate a clinically relevant structure. Another approach is 3D bioprinting, and we recently acquired a 3D printer in our lab, which instead of using like a 3D printer that uses polymers or plastic, our ink is a bio ink composed of cells and collagen, and we can now actually print, take a CT of a patient, load the printer with cells and extracellular matrix. Here we're downscaling it in size, but we can see that we printed something that looks like a left atrium based on a CT of a patient. And we can actually induce something like the left atrial flutter in this model. So we're getting more and more to try to generate an atrial-like structure. What about the ventricle? Here we're using another technique to try to generate a scar model of VT. In this technique, we are using a material that we coat the culture plate, and this material prevents attachment of cells or extracellular matrix. You then add a photoinitiator together with a material that when hit by a laser, it ablates this material and exposes the bottom of the dish. So now you can load an image that you want to print to micro pattern on the tissue, and then use this laser approach, UV laser approach, to generate a micro pattern where you remove the anti-fouling material, the material that prevents attachment of cells. And then when you see the cells, you can actually generate exactly the image that you wanted to print. So here we wanted to make a very gross cartoon-like structure of the infarct with an area of normal tissue and then a narrow conducting strand, which you can see here. So we were actually able to print and get exactly this structure. These are all cells that are printed exactly in the way you want, microscopically. And now we can study different properties. You can see that this is how it looks microscopically. You see the cells in the narrow area. And you can see that we can now pace. Interestingly, it was very difficult to generate re-entry in this model. And it turns out that we can only generate re-entry when we caused local conduction abnormalities by generating something like a abnormal structure within this strand. So it's not enough to have an homogeneous strand. You need to have some kind of abnormality, as you can see here. And this allows the generation of unique conduction directional block and development of VT, as you can see here. Okay, so we can generate an arrhythmia. So we're really studying what allows us to generate conduction block, and there's a lot of work that we are doing on this. We can actually now go further and actually model a specific scar, either from pathology or from MRI. So if you have this image, you can digitize it and use the laser micropatterning to, as you can see here, these are the cells, to see the cells exactly like the pattern, the microscopic pattern that you wanted to generate. So we can now print an infarct border zone, for example, and then look at conduction in this infarct border zone. And you can see, for example, that we could generate a VT circuit that is identical to the one that was generated in this model. Okay, so I told you about how we are moving from simple single cell model to 2D tissue models to 3D models. What about interventions? Can we manipulate the activity of the electrical activity? And one of the problems in electrophysiology that we don't have any way to really reversibly intervene in our system or manipulate our system. So when we do ablations, this is irreversible. Once we do it, we cannot assess the same model again. Similarly, when we give drugs, it affects the entire tissue, not a specific area within the tissue that you want to study. So I want to take you to another technology that won Nature Method of the Year, which is called optogenetics. Optogenetics started when scientists realized that algae move away or toward light because of the presence of light-sensitive channels, with the prototype being channelrhodopsin, which is a light-sensitive cationic channel that opens following exposure to blue light and allows mainly sodium ions to enter the cell. Then scientists understood the potential of this approach, actually in Stanford, and they cloned it and then expressed it in neurons in vitro or in different animal models. And this really transformed the way neuroscience is done. This is a very famous movie in which channelrhodopsin was expressed in the brain of this mouse. You can see that the mouse walks happily, and then there's an optical fiber in its brain, and you can see that when illuminated, the animal starts to walk in circles. This is because they are illuminating only one side of the motor cortex. So this really changed the neuroscience, and we wanted to see whether we could do the same in the heart. And again, I apologize because I have the feeling that this movie will not project well. So what we did was use adenoviral gene delivery approach to generate a gene therapy-like strategy in which we can deliver the channelrhodopsin gene to the heart. And once the cell starts to express the protein, we can then try to modulate electrical activity with light. And our first approach was to try to generate a light-induced pacemaker. So this is in the Langendorf mode. I see that it's really not moving, but what you should see is that the light, every flash of blue light should pace the heart. So you can see in this slide, in the ECG of the heart, that we can actually pace the heart with light at increasing heart rates. So like an electronic pacemaker, we can pace the heart with any rate we want. We can do this while the heart is still beating in the chest. Again, it's beating. I don't know if it projects, but again, we're flashing a lot of flashes of blue light, and we can pace it in any way we want. So we generated a light-induced pacemaker. We then moved on to see if we could do more than that. So we thought about perhaps we can use it as a better CRT approach. You know that with CRT, we are limited with the number and location of the electrodes that we use, and we don't always get optimal results. So the question is, can you improve this approach by having the ability to pace the heart from multiple sites, or actually from indefinite number of sites? And to test this, we generated a model in rats in which we injected the channelrhodopsin to 3 areas in the ventricle, 3 or 4 areas. And we then studied using optical mapping the activation. This is sinus rhythm. You can see that sinus rhythm is very fast. The entire ventricle is activated simultaneously. To model the synchrony, we pace the right ventricle. And then you can see that it takes much longer to activate the left ventricle. And then we illuminated the 3 sites, and this allowed to synchronize again the electrical activity in the heart. So this summarizes the study. Sinus rhythm is very fast. It takes very short activation time to activate the ventricle. When we pace the right ventricle to simulate left bundle branch block, we can see that activation time of the left ventricle rises. And when we employ multi-site optical pacing, it goes back to the normal activation sequence. So we can actually homogenize activation in this model. And we now have studies in different models that this results in mechanical improvement. We can now improve this significantly. In that study, we controlled where activation starts from by where we injected the cells. But of course, we can use different illumination patterns to generate any activation sequence we want. So in this model, we can now activate the cardiac tissue either from one site, as you can see here, from 4 circles, as you can see here, from a medium rectangular, as you can see here, or diffuse spacing, and study the different effect on conduction, different conduction activation times on mechanics, on electrical activities, on arrhythmias, and so on. Can we do the opposite? Can we stop electrical activity? And we can do this by using light-sensitive channels that either cause hyperpolarization or continuous depolarization. This can allow us to stop electrical activity. So we can think of a defibrillation approach. You can see a rotor in which we shine light and we stop its electrical activity. More recently, we designed a very nice potassium-based light-sensitive channel, which can robustly suppress electrical activity, as you can see here. And this is an interesting channel in which a short stimulation results in a few seconds of silencing of activity. So now we have a new tool in arrhythmia in which we can silence specific areas in the tissue at will. Here, for example, we can study and generate conduction block. So this is our tissue model. It still doesn't project very well, but you can see that we generated a conduction block here. We can generate a conduction block at another site by light. Okay, so we generate a functional conduction block wherever you want. And we can also do this to try to defibrillate an arrhythmia. This is an arrhythmia that we generate in a dish. We shine light and it stopped activity. We then can see which areas are critical for the perpetuation of the arrhythmia, shine light on a specific area. Here, for example, we shine light only on the center. You can see that it suppressed activity, but the periphery was enough to keep the arrhythmia going. And we can do this many ways. For example, trying to do like a maze procedure, and you can see what is the minimal tissue needed to sustain an arrhythmia. Here, for example, we are doing a crisscross. You can see that this terminates the arrhythmia because all areas are not enough to sustain the arrhythmia. And finally, we wanted to see we defibrillate an intact heart. So in this case, we did an infarct model to generate a VT model. When we created the infarct, we also delivered by adeno-associated virus the light-sensitive channel into the infarct border zone. We then took out the heart, and now we will try to induce VT-VF outside the body. So we can see that now we are pacing, or you see in a second, pacing the heart. We're creating ventricular fibrillation. You can see that the heart is fibrillating. We will now shine light on the heart, and this terminates the arrhythmia and restores sinus rhythm, as you can see in this ECG. So with that, I would like to end. I hope I'm in time. Thank the people in my lab that did this work. This is all my students. This is a picture of Haifa. This is how we live in Haifa. Everybody has these kind of houses. And I'd like to thank our collaborators all over the world. And thank you for your attention.
Thank you so much for that awesome tour de force talk, Dr. Gepstein. I'll open it up to any questions. Feel free to turn on your camera, ask any questions, or type it in the chat box.
I can start it off. Lior, fantastic talk as always. it seems like there are two kind of major next steps to be done. One, and I'm sure you've thought a lot about both. One is engineering or constructing a whole heart, which on the one hand seems like, well, that's crazy complicated. You have the nerve cells, the fibroblasts, et cetera. On the other hand, that's what our DNA was designed to do. So you wonder if it's just unlocking certain things and it should kind of take care of itself. So curious and it's just the right concoction of various things. Of course, it's outside the normal environment of the entire body, but curious what your thoughts are on next steps there, major barriers there, or if we're getting closer to that.
So in the field of tissue engineering, there are like two approaches. One approach is called organoids, in which the idea is, like you said, our organs know how to generate these complex structures of different cells. They know how to So organoids, you try to do self-organizations of cells to generate something that will look like the native tissue. And it works very well from some tissues like the intestine, brain to a certain extent, and other tissues. In the heart, it's progressing, but you don't get extremely complex something that looks like the heart. But again, people are working on it and it's progressing. The idea is that if you bring the right precursor cells, they will self-organize in the appropriate way. The second approach is to help this happen, is to generate the right type of cells and then to put them in place in the right place. And this is where the 3D bioprinting enters. And so these are the two broadest ways how to do it. 3D bioprinting just starts. I'm not sure we still have the tools. For example, if you only think of the vasculature, you want to generate the entire artery from a big artery, like a coronary artery, to the small arterioles, and so on. And then you need all the correct fiber orientation of the cardiomyocytes and the nerves and so on. But I think eventually a mixture of some kind of self-organization that occurs together with some kind of triggers to allow the cells, to become in the right place will come up. And we're gradually getting there. I mean, initially, we never thought that we can make all the different, Lego parts of the heart. So now we can make them. Now we need just to put them in the right places. And it will take slow, but eventually this will also come.
The other thing that seems like it's been lagging is the delivery. And so there are all these incredible technologies, CRISPR, all of the great work you've done with iPS cells, but we're still, it seems, reliant on this fairly primitive way to localize your target with an injection, for example. So curious also about what the forefront of that is and how you think about that.
So we're doing, both gene therapy and cell therapy, and gene therapy and CRISPR has been remarkably successful in the liver, for example. The liver is a great target for all types of gene therapy, as well as stem cells that you harvest, manipulate them ex vivo, then return them to the body. The heart has been a difficult target for this, but again, it's progressing. And after, most of the companies are looking on the liver and so on, but now more and more people are starting to look at the heart because it's more difficult, but it's a very attractive target. Regarding cells and tissues, it's been slow. There's now 2 clinical trials or 3 clinical trials going on in transplanting cells, heart cells generated from iPS cells or embryonic stem cells. 2 of them are using tissue patches, like I told you, and one of them is injecting cells. We'll see how it goes. Again, it's unlike other areas where it's progressing much faster, like macular degeneration, where you don't need a lot of cells and you cannot make not do a lot of harm. In the heart, you can do harm if you generate arrhythmias and things like this. You can cause serious, so everything is slower in the heart. But in general, delivery, I agree. Delivery is a big barrier in the heart.
I mean, it seems like
everything is
that's
growing, but delivery has not progressed to the same extent.
it seems like there needs to be a kind of a paradigm shift in thinking, less about like the injection that might work for an eye and how to really engineer, integration in a slicker way than simple injection.
Yeah, but I mean, eventually it will come. Takes long. I've been working in this for 2 decades. I'm a patient guy.
All right, thanks. Awesome talk, Lior.
Any other questions? I know it's 1 p.m. now, so if people have to leave, that's okay. But wanted to thank you again for taking the time and joining us for Grand Rounds, and hope to see you again soon here at UCSF, hopefully in person sometime.
Great.
Yeah. All right. Thank you,
Bye-bye.
everyone.
Thanks, Lior.
Hi, Lior. I don't know,
Hey,
you were in the
Ivy,
lab
how are
like
you?
15 years ago.
You look the
Something
same.
like that.
You
I just
haven't
wanted to say
changed a
hi.
bit.
Good talk.
Thank you. Yeah, some of this started with you.